Welcome back to our another video tutorial. This is Rajesh. Welcome to RS biology tutorial. In this session, we are going to be discuss about derivation of DNA structure in the chapter of molecular basis of inheritance. Right? Derivation of DNA structure. Yes, so already we have discussed about the polynucleotide chain structure. Which type of bonds are present in the polynucleotide chain? There is 4 type of bonds: N-glycosidic bonds, phosphoester bonds, phosphodiester bonds, phospho and hydrogen bonds, etc. Now, in this session, actually we need to discuss based on which studies, based on the studies of which scientists, the DNA structure is described actually. We know the double helical structure of DNA described by the famous scientist Watson and Crick. But before the Watson and Crick, there is another scientists done the important work to describe the DNA structure. That only we are discussing now here, right? In this session actually, first method is X-ray diffraction method. X-ray diffraction method. This X-ray diffraction method is described by the scientist Maurice Wilkins and Rosalind Franklin. Maurice Wilkins and Rosalind Franklin obtained a very fine X-ray diffraction pictures. With the help of some X-rays, they obtain some particular pictures of the DNA. This method is called as X-ray diffraction pictures method. This method is developed by which scientists? Maurice Wilkins and Rosalind Franklin. Yes, it was suggested that the length or pitch of 1 ton of DNA is 3.4 nanometers. Right? What is the ton? What is the pitch? We will discuss later at the time of discussion of DNA structure actually. But see, based on the studies of Maurice Wilkins and Rosalind Franklin, what they suggested that the length or pitch of the 1 ton of the DNA is 3.4 nanometers. Okay, this is about X-ray diffraction pictures method developed by Maurice Wilkins and Rosalind Franklin. Based on these studies also, the DNA is described by the Watson and Crick. Means Watson and Crick depends on the study of Maurice Wilkins and Rosalind Franklin to describe the DNA structure. Okay. The next one is, next scientist name is Erwin Chargaff. He was play very important role in the description of the DNA structure. What their rules here? Actually, what function they done? Erwin Chargaff performed the base composition studies. Nothing but base pairing. They are simply given 1:1 ratio. What is base pairing? What is 1:1 ratio? Suppose see, if one strand having adenine, the opposite strand should having thymine. That means one purine. If one strand having one purine, the opposite strand should having one pyrimidine. This is called 1:1 ratio. One purine, one pyrimidine, or one pyrimidine, one purine. This is called as what? Base pairing. This 1:1 ratio is described by which scientist? Erwin Chargaff. Okay. And the rules only applicable to double stranded DNA but not to single stranded DNA. Yes, always you must remember, Erwin Chargaff's rules are only applicable to double stranded DNA. Why? Because in double stranded DNA only base pairs are present here, base pairs. But in single stranded DNA there is no base pairs. That's why the Erwin Chargaff's rules are not applicable to single stranded DNA. They are only applicable to double stranded DNA. Right. Once see, what are the rules proposed by the Erwin Chargaff? Now, rules of Erwin Chargaff. Suppose it is first rule. In any double stranded DNA, purines and pyrimidines occurs in equal amounts in one double stranded DNA molecule. Suppose in any double stranded DNA molecule, the amount of purines equal to amount of pyrimidines. This is the first rule of Erwin Chargaff. Right? See here, second rule. Total number of purines equal to total number of pyrimidines. Total number of pyrimidines. Okay. In any double stranded DNA, total number of purine, that means A plus G, equal to total number of pyrimidines, that means T plus C. See here, total number of purines in the DNA, wherever A is there, wherever there, total A and G counted here, total T and C counted here, both values should be same here. Okay. And third point is, the ratio of purines and pyrimidines is always 1 or constant. Suppose see, total number of purines, ratio between total number of purines and total number of pyrimidines always 1. It is constant for all species. In each species same value will be there. Okay. A plus G by T plus C equal to 1. It is constant for all species. Right. Suppose see, this is the one double stranded DNA. This is the one double stranded DNA. Once we have to count here, what is the total number of A plus G here? Suppose A, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Here A plus G are 13. By T plus C, how many T plus C are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Yes, T plus C also 13. 13 by 13. Suppose we can take the example of this DNA. It is one strand, it is another strand. There is nitrogen bases. Okay. Yes, that should be correct here. The total number of purines divided by total number of pyrimidines equal to 1. The ratio between total number of purines equal to total number of purines and total number of pyrimidines is always 1. Next, base ratio. What is the base ratio? Yes, A plus T by G plus C. Here A plus G by T plus C, but here not like that. A plus T by G plus C. It is specific for species. It is not same in all species. Not same. Okay. Suppose A plus T. How many A plus T's are there here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Here 16 A plus T are there, and G plus C. How many G plus C are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 16 by 10. Base ratio, for example, we can take in this DNA. What is the base ratio? It is 16 by 10. A plus T by G plus C. That means 16 by 10. It is not equal to 1. It is different. It is different in all species. Specific in each and every species here. Okay. Suppose examples are given. The base ratio of prokaryotes, especially in E. coli, it is always 0.92, less than 1. But the base ratio of eukaryotes, suppose in humans, the base ratio is 1.52. That means it is more than 1. In prokaryotes, the base ratio is less than 1. In eukaryotes, the base ratio should be more than 1. Okay, very simple. Yes, what lines are given here? The two DNA strands are complementary to each other. What is the complementarity? Once we have to discuss here. Complementary to each other. And therefore, if the sequence of bases in one strand is known, then the sequence in other strand can be predicted. Suppose it is a one double stranded DNA. Suppose we know the sequence of this strand only. We don't know the sequence of this strand. But here based on the sequence of this strand, we can easily predict the sequence of opposite strand. This is the complementarity. Why? Because we know the rule, always A bind with T, the same way T bind with A. Always G bind with C, in the same way C bind with G. So based on this rule, Based on this rule, we can easily predict the sequence of opposite strand with the help of the sequence of one strand. Okay, if the sequence of one strand is known, we can easily predict the sequence of opposite strand of DNA. This is called complementarity. Okay, yes, this is about derivation of DNA structure and Chargaff rules here. Based on these two methods. What is the first method? X-ray diffraction method, and another one is what? Rules of Chargaff. Based on these two methods, the Watson and Crick proposed the very famous double helical structure model of DNA here. Okay, about that we will discuss in the next topic. I hope to understand the concept. We will meet in the next video. Thank you. If you got any doubt, you can ask the doubts in the comment section. I will try to clear every doubt. Thank you.